I'm going to be talking to you today about modulating inflammation through some different mechanisms. So I'm going to be talking today mainly about nuclear factor kappa B. Before I make a start, I'll do what I always do and just sort of do a quick sound check. So I'm assuming that, or I'm hoping that you can all hear me nice and clearly, otherwise I'll tweak my volume or whatever. So there's a small chat box down in the left-hand side of the corner. Great, I've got Michelle is typing away there. Great, and some other people. So super, right, okay. So we treated ourselves to a nice little lead the other day so that we can plug it directly into the IT system. So hopefully we'll have no issues with our sort of sound quality or anything like that. So great, so I'm going to kick off. So as I said, today I'm going to be looking at different ways of modulating inflammation and having a look at not only our sort of favorite fatty acids, but also at some sort of other areas, other nutrients and other potential sort of supplements. We're doing a lot of R&D here at iGenis, and so we're looking at some new product ideas to complement our wonderful range of omega-3 supplements. And for those of you who do know us, you'll know that we are the lipid experts. Our wonderful range of products really kind of centers on high-strength EPA for targeting inflammatory control. However, inflammation can be regulated in a number of very different ways. And whilst we kind of center on the role primarily of omega-3 and their individual roles in regulating inflammation and inflammatory resolumics, there are a number of other pathways that we can kind of look at. We know that omega-3 and omega-6 primarily centering on EPA, DHA and AA act as structural components of our cell membranes, and they influence cell membrane integrity by influencing the fluidity. We know that the ratio of these fats have both structural and functional consequences, and that most importantly our requirements for omega-3 are often not met, increasing our risk of developing a number of inflammatory associated conditions. So our omega-6 and our omega-3 sit in our phospholipids within our cell membranes, and any changes in cell membrane fluidity can alter the important structural functions such as lipid rafts and caveoli formation, which then has a knock-on effect by altering the localization of various proteins, such as those that are involved in cell signaling, for example, but also those that are sort of involved, sort of like ion channels, for example, that are required for functions that require the transport of sodium, potassium, calcium, and so on. And we have all these kind of fatty acid transporters. We have a variety of membrane proteins that are also embedded into our cell membrane that have to be able to sort of float around and interact in a sort of fluid way to be able to act normally, as you would expect in a sort of healthy state. So if we just kind of have a quick sort of look at an example really of how omega-3s and their wonderful way of sort of modulating inflammatory, the sort of fluid mosaic model, as you were. One of the sort of wonderful ways of sort of explaining their role in cell membrane fluidity is how they help to sort of regulate insulin signaling, for example. So if we have a little look at our diagram here, we know that under conditions where there is low insulin, most of the GLUT4 transporters that are required for glucose transport are sequestered in these little intracellular vesicles within muscles and fat cells. And when insulin is present, it induces a rapid increase in the uptake of glucose by inducing the translocation of GLUT4 into the cell surface from the vesicles within the plasma membrane. Dietary omega-3 within the cell membrane itself actually help to increase insulin binding by changing the fatty acid composition of the phospholipids surrounding the insulin receptor. And it's this mechanism that dietary fatty acids are known to modify insulin action. So if the cell membrane isn't fluid enough, then we get changes in the insulin receptor, and then it's not able to recognize in the insulin molecule itself. And so one of the key functions of omega-3 come from the sort of downstream products that are derived. And if we just go back to our older picture, so we know that they sort of have this role in cell membrane fluidity, but they also are involved in the production of sort of eicosanoids and docosanoids and pro-resolving products. In order to do that, they have to be released from the cell membrane, mainly by phospholipase A2, for example, where they enter into the cell itself, and they then are able to become converted into these sort of downstream molecules. So the focus on there was really to sort of highlight the fact that they need to be released, and if they need to be released, we need to be able to ensure that we maintain adequate levels of omega-3 within our cell membranes themselves. So we have to have nice full stores, if you like. And a lot of the focus has been sort of quite recently placed on the importance of maintaining adequate levels of omega-3. Certainly coming from the science community, it's suggested that we should be aiming to achieve an omega-3 index of at least 8%. And the reason that I'm kind of going down this line at the moment is that a lot of the recent sort of meta-analysis that have been published have been clouding a lot of the opinions surrounding the efficacy of omega-3 benefits. But these often report intakes that are relatively low, either below 1 gram or around the sort of 1 gram mark. And this paper by Ruck was quite interesting. He was kind of highlighting the consequences, if you like, of omega-3 dosing at around about 1 gram. And in his study, he showed that you can increase the omega-3 index, but in this population, the mean index of 3.6 only went up to about 5.4, with about 16% of subjects actually achieving an EPA DHA level of less than 4.8%. He also showed that the AA to EPA ratio was reduced from 20 to 7.2, which is not the sort of ideal less than sort of 1.5 that has sort of arisen from a lot of the JELIS studies. Clinical relevance really is that it's not until you achieve an omega-3 index of at least 5% that you get this sort of dramatic reduction in sort of coronary death that is observed. And so there's a nice sort of little cutting here that I've taken from the study, and they're really kind of highlighting the importance of monitoring EPA and DHA levels in patients, and so that we can identify those that are at increased risk of not just sort of sudden cardiac death here, but sort of health conditions as a whole, and that maybe changes, or looking at the changes in EPA and DHA incorporation, should be monitored so that we can sort of assess whether a required EPA DHA level has actually been reached in a particular patient. So there's still this kind of increasing importance being sort of focused at getting our levels up to a certain amount. So basically, yeah, so we need our cell membranes to be packed nicely with EPA and DHA to ensure that we have adequate supplies to be able to sort of produce the fatty acids that are required for all these different pathways. Right, okay, so one of the areas of inflammation in sort of inflammatory regulation that we've not really covered in some of our previous webinars is via a number of different signaling cascades, such as those that are involved in regulating transcription factors such as nuclear factor kappa B, which will be kind of most of the sort of focus of today's talk. And then we've got sort of active protein A1, We've got various recognition proteins like the toll-like 4 receptors that are embedded in cell membranes. And then we go down to sort of the peroxin proliferator activated receptors, the PARs, that they're here, that are involved in regulating things like glucose and lipid metabolism, but they actually also interact with different other sort of transcription factors to repress pro-inflammatory genes, for example. So we've got all these different sort of other pathways that we don't really pay that much attention to, or we haven't, that we're going to sort of maybe start focusing on and elaborating on. So as I've said, inflammation is complex. It involves various sort of multi-systems that affect a wide range of cells and tissues. And there are numerous transcription factors that are involved in regulating many of the sort of mediators, such as sort of cell adhesion molecules. We've got pro-inflammatory cytokines, we've got our chemokines, we've got various sort of reactive oxygen species. And all of these are involved in the generation and sort of propagation of the inflammatory response itself. And all of these are under the control of various transcription factors, such as transcription factor activator protein A1 and specifically nuclear factor kappa B. And these two play roles in the expression of many of the genes whose activity and expression tend to be elevated in inflammatory conditions. Making them targets for not only a lot of sort of pharmaceutical interventions, but certainly sort of from a nutraceutical perspective, we can start looking at sort of diet and dietary factors that can help sort of regulate a lot of the transcription factors that are involved in producing all these sort of byproducts, if you like. And these transcription factors regulate gene expression in response to a variety of stimuli. And these include things like pro-inflammatory cytokines. There are numerous growth factors. You've then got sort of stress response, as well as a number of sort of bacterial and viral infections as well. So if we look at nuclear factor kappa B, this is normally, or nuclear factor kappa B is normally located in the cytoplasm of the cell, and it's in an inactive state because it's complexed with this sort of like little inhibitory protein called IKB alpha. Through a variety of extracellular signals, activation of the enzyme IKB kinase, or IKK, phosphorylates this protein, then resulting in dissociation of our nuclear factor kappa B, which then is allowed to sort of move down into the nucleus, where it sort of combines with sort of DNA, transcribes downstream DNA into messenger RNA, which is then obviously translated into proteins that are involved in various sorts of changes in cell function. So we're looking at sort of regulation of anything through from sort of COX enzymes through to cytokines, chemokines, and so on. So nuclear factor kappa B is one of the principal inducible transcription factors that modulate a trigger of a cascade of signaling events that are involved in integrated sequence of various protein-regulated steps. Some of them, as I said, are sort of key targets for intervention when we're thinking about sort of treating inflammatory conditions. And this sort of typical pathway is triggered by a number of microbial products via the toll-like 4 microbial pattern recognition receptors, which I'll talk about in a moment, and primarily with pro-inflammatory cytokines such as TNF-alpha and interleukin-1. And these two are actually the sort of the archetypal pro-inflammatory cytokines that tend to be rapidly released on tissue injury or infection and sort of interact with various sort of transcription factors and pathways that then sort of increase this whole inflammatory response. However, there are a number of endogenous ligands that can also trigger these toll-like 4 receptors during tissue injury in certain disease states, which also act to promote inflammation without the sort of presence, if you like, of an infection. And we also have sort of other factors that also trigger toll-like 4 receptors, which I'll talk about as well. But the sort of focus really is on nuclear factor kappa B as widely implicated inflammatory diseases. And there's a lot of intention, as I've said, on focusing on the development of sort of anti-inflammatory drugs and anti-inflammatory sort of nutraceuticals that target nuclear factor kappa B. So, so we've got various triggers. So we're looking at things like increased levels of oxidative stress. We've got our pro-inflammatory cytokines. Obviously, we've got bacteria and viruses. A lot of physical, and we also know sort of a number of sort of psychological stress factors can induce nuclear factor kappa B. And in fact, the whole sort of interaction of psychological stress and the progress of depression, or the risk of depression, has also been linked with toll-like 4 receptors and sort of bacterial types of bacteria that then link through and stimulate nuclear factor kappa B. And again, I'll speak about that in a minute. And we've then got sort of things like growth factors. We've got DNA damage, so the sort of apoptotic cell, if you like, sort of shutting down and dying, can also trigger this sort of inflammatory response that centers through the activation of nuclear factor kappa B. So this sort of godfather, if you like, of the sort of transcription factors then sort of gives rise to, or has the potential to give rise to, sort of pro-inflammatory cytokines, chemokines, products that regulate sort of apoptosis, differentiation, cell cycle, and again a number of growth factors, immune receptors, and antioxidants and stress response genes as well. So he's a bit of a powerful guy, is nuclear factor kappa B. One particular area of interest when it comes to sort of regulating and using it as a target for sort of intervention is how nuclear factor kappa B plays a role in carcinogenesis. So it sort of starts to really kind of open up here when we start looking at some of the genes that nuclear factor kappa B has the ability to sort of switch on or switch off. Those that regulate inflammation, those that are involved in sort of apoptosis or sort of anti-cell survival. We've got those that are involved in proliferation, metastasis, angiogenesis, tumor promotion. So it's not surprising that we're seeing a lot more sort of focus on regulating some of the pathways that are involved. So I've very briefly before mentioned the toll-like receptors. So these are a family of membrane-bound sensors that sit there, and their primary focus is to sort of detect and respond to microbial infections. And this sort of different family that each one within the family recognizes a different sort of area. So we've got some that respond specifically to things like bacterial DNA, others to viral double-stranded RNA. But the one that I'm going to focus on today is toll-like receptor 4, which responds or is activated mainly by lipopolysaccharides. And so the lipopolysaccharides are sort of carbohydrate chains that are found primarily on gram-negative bacteria. Activation of toll-like 4 culminates in the activation of our friend nuclear factor kappa B, which again goes off and does all these kind of pro-inflammatory cytokine gene expression initiation of. Toll-like 4 agonists include, as I've said, gram-negative bacteria. So I'll be talking in a minute about the importance of different bacterial species in sort of initiating sort of inflammation and how they interact with the gut membrane themselves to sort of influence the process of sort of leaky gut, if you like. But we also know that there is a big role of saturated fats, and specifically high-fat diets that are rich in palmitic acid and stearic acid, that are known to elevates levels of lipopolysaccharides that are then directly related to this increased intestinal permeability. And the sort of pathways that are involved is sort of reduced expression and activity of various tight junction proteins such as zonula and occludin. These sort of effectively sort of act like the glue that holds the sort of gut epithelial cells together to create a barrier that separates the intestinal lumen and the bacterial population. When we start to get breakdown in these tight junctions, we are then allowing sort of not only harmful bacteria, but also we get this sort of influx of lipopolysaccharides, which are then free to initiate this sort of cascade of inflammatory processes through nuclear factor kappa B pathway. So, as you're probably all aware, there's quite a lot of sort of voice at the moment in the media about sort of like telling everybody that we should be eating much higher fat diets. And I think some of the public messages need to be a little bit careful, because I think we've made so many mistakes in the past with sort of, sort of low fat diets, that to then sort of go sort of crashing in, sort of telling everybody to eat high fat diet needs to be a little bit cautious, and I think a little bit more information to people on what we mean by high fat diets. So again, if you've sort of attended any of our sort of previous webinars, we really kind of focus on sort of whole foods and organic, and the importance of things like grass-fed products to ensure that we're getting a nice balance of sort of good fats to bad fats, as it were. And this is just a slide that I've included to sort of show the difference between certain fatty acids that are present in sort of grass-fed products and grain-fed products. So I've really just sort of isolated some of the fatty acids out here. So really, it's just to sort of highlight the fact that if you eat grass-fed products, then your intake of sort of these inflammatory saturated acids such as palmitic and stearic are going to be much lower, and your intake of EPA, DPA and DHA, which are anti-inflammatory omega-3s with a multitude of health benefits, are going to be much higher. So it's about the focus on really kind of limiting our intake of certain fats, but not sort of going down the sort of just sort of high fat diet without some sort of knowledge of what it is that we're actually eating. So when it comes to looking at sort of gut dysbiosis, that is then linked through to gut permeability. So we've got all these sort of factors that we know are involved, anything through to sort of poor liver function, which can increase various levels of inflammation. But we know that, and again, this is another area that's being highlighted quite heavily in the news, is our overuse of antibiotics, which is having a sort of detrimental impact on our gut function through modulation of our sort of ratio of bad bacteria to good bacteria. We've then got sort of various aspects of diet which are also known to contribute to gut dysbiosis. So not just sort of high fat diets, but also sort of diets that are low in complex carbohydrates. So sort of adding to the sort of low dietary fiber sort of load to the diet. So our good bacteria like to thrive on complex carbohydrates to produce all the lovely sort of health protective byproduct that sits and look after our gut health. But anyway, so when we sort of start looking at gut dysbiosis, which can then sort of lead to this sort of gut permeability with sort of excessive amounts of lipopolysaccharides, we've got then sort of other sort of areas that I've discussed before, the sort of endorphins. We've got our issues with gluten and casein, sort of opiates that all help to sort of increase gut inflammation, which are then going to sort of trigger back and tie back and sort of contribute to sort of whole body inflammation. So sort of having a look at these areas and getting our gut function healthy and our gut population healthy is really, really important. So as I've kind of mentioned, that the pro-inflammatory effects of what we consider to be sort of high-fat diet has mainly been attributed to sort of the inflammatory properties of specific dietary fats. So sort of rather than high-fat diet, specific fats. So we should be sort of looking at sort of increasing more polyunsaturated fats, monounsaturated fats, and limiting a little bit of the saturated fats. We know that fatty acids trigger this sort of inflammatory response by activating, or sort of acting, sorry, via our toll-like 4 receptors, signaling through in adipocytes and macrophages, which contribute to the inflammation within adipose tissue in itself. High-fat diets appear to affect the composition of the gut flora, and we see an increase in firmicutes. You have to pronounce, forgive my pronunciation here. So firmicutes, sorry, and a decrease in bacteroidetes. So it's this balance between the two sort of areas of bacteria that is really important. So excess dietary fat not only increases sort of its systemic exposure to potentially pro-inflammatory free fatty acids, but it also facilitates the development of something called metabolic endotoxemia, which is this increase in plasma lipopolysaccharides, which then triggers this systemic low-grade inflammatory response within a range of different tissues. And my little slide here sort of like kind of sums it up quite nicely, really. So high-fat diets alter our gut microbe composition. We get an increase in glucagon-like 2 peptide, peptide 2, which is involved in sort of regulating gut permeability. If you increase glucagon-like peptide 2, then you get a lower gut permeability. We get an increase in lipopolysaccharides from our firmicutes, which then contribute to an increase in lipopolysaccharides, which activate toll-like 4 receptors, upregulate the expression of pro-inflammatory cytokines. And as obesity is considered to be a chronic inflammatory disease, we get an increase in production of pro-inflammatory cytokines that trigger this inflammatory response that ultimately leads to weight gain. And then we've got another pathway here. So our intestinal microbes alter by high-fat diet, suppress something called fasting-induced adipose factor expression, which promotes lipoprotein lipase, which is a regulatory enzyme that enhances the absorption of fatty acids. We get a build-up of adipocyte triglycerides, thereby increasing fat storage, leading to obesity. So again, there's a lot of interest in the media at the moment within the science community of modulating body composition through probiotics. And that is an area that we're kind of like looking at the moment in regards to specific microbial strains and their role in different sort of areas of health. Okay, so I'm not going to sit here and sort of read out all these different bacterial strains because my pronunciation is rather poor. But we do know that there are certain strains of bacteria that do very specific function. And so if we're looking at different probiotics for different areas of health, we can see that some are known to help with sort of membrane integrity within the gut cells themselves, or the sort of colonic mucosa itself. Those that help to sort of repair leaky gut, for example. Some reduce candida numbers, reduce gut wall permeability. Others are meant to be antimicrobials, that therefore are going to inhibit the growth of some of the more harmful pathogens. And others are known to ferment sugars into lactic acid, which increases the acidity of the intestine, which is rather good because that helps to inhibit the reproduction of some of the sort of harmful microbes. Some of the more harmful microbes like an alkaline environment. So sort of looking a little bit at the evidence of different various sort of bacterial strains, we know that, your sort of, home commensal bacteria, gut flora, and several probiotic strains can directly modulate the amount of tight junction proteins at cell boundaries. This can then obviously therefore help to either prevent or reverse adverse effects of pathogens. And again, there are several probiotic strains that have shown to have beneficial impacts on tight junctions and intestinal barrier functions by increasing various proteins, zonula, occludin, and so on. And you can see this through a decreased sort of fecal content of these proteins, which are generally a marker that indicates that there is enhanced gut permeability. And another sort of side effect, if you like, is that if you pre-treat with probiotics, or if you treat with probiotics, you get a direct decrease in pro-inflammatory cytokines, which is always going to be slightly helpful. Another sort of aspect of certain bacterial strains is that we can sort of divide them into those that like an acidic environment and those that like an alkaline environment. Different bacteria have different functions and have different outcomes. Some have been sort of associated with sort of an increase in colonic tumor growth rates, for example. Others are known to prevent tumor formation, and a lot of it is all about how they affect sort of the fecal pH. So some tend to be ammonia producers, those that predominantly sort of feed off some of the sort of protein that makes it through into the colon, for example. Others can sort of decrease some of the sort of bile acid. These are sort of toxic sort of secondary bile acids, and others decrease some of the sort of products known to be quite toxic that sort of end up in our colons. And others have direct sort of benefits for sort of immune system function as well. And just to sort of highlight the different sort of roles and of different bacteria, if we look at sort of colon function, for example, so the sort of byproducts of the food that we eat, primarily talking about protein and carbohydrate here. So anything that sort of enters the cecum of the ascending colon, this is the sort of area, the main site of carbohydrate fermentation. So all your sort of good bacteria that are sort of creating things like butyrate and acetate that are going to be beneficial to sort of not only to the bacteria themselves, but also going to be providing fuel directly to keep our colonocytes, the cells within our colon, nice and healthy and ticking over nicely. Butyrate is also known to be a tumor suppressor. So the bacteria that sort of sit within our ascending colon tend to like to sort of munch away on all this sort of carbohydrates. So we're talking about sort of complex carbohydrates and fiber and things like that. So if you've got a low fiber diet, there's not going to be much for your sort of colonic bacteria to sort of eat, and they if they don't get fed, then we start to see a sort of change in the composition of different sort of strains of bacteria, for example. As we move across into the transverse colon and down the descending colon, this is the main site of protein fermentation. So this is kind of a lot of sort of sulfur producing bacterial strains tend to sit within this kind of area. They tend to give rise to end products that are not quite so kind to our colon as the byproducts of sort of carbohydrate fermentation. And so we start to see sort of a shift in the sort of acidic environment to the alkaline environment. So various sort of things that we could do dietary-wise to sort of keep our colon and our sort of gut function ticking over nicely. So this sort of shift in balance between sort of healthy and sort of non-healthy sort of bacterial state, if you like. We know that we get this sort of shift from Firmicutes to Bacteroides in a wide range of sort of health conditions. As we age, for example, we sort of generally sort of get less beneficial sort of gut flora, but there's various sort of diseases that are also associated with this sort of dysbiosis, anything through from sort of irritable bowel disease to, as I've sort of said already, sort of obesity, metabolic syndrome, type 2 diabetes. And it's this sort of change in the ratio of these sort of bacterial strains here that seems to be the key. So when it comes to sort of improving gut health, we know that there are specific strains of probiotics that have been shown to be sort of beneficial in gut function. And within those, there are particular strains that I've sort of mentioned in a couple of slides back that are also known to have very specific roles in areas of sort of gut function, in sort of tight junctions and things like that. But the importance shouldn't be underestimated as well of our sort of short chain, non-digestible carbohydrates. And so high dose inulin, for example, sort of taking sort of 10, 20 grams of inulin a day is known to give us a nice shift, if you like, of sort of bad bacteria to good bacteria. So if we start feeding our good bacteria, and then we can sort of slowly sort of persuade those to sort of overcome and help colonize those. So again, there are a number of really beneficial prebiotics out there that are naturally occurring, found in cereals, fruits and vegetables, and they tend to target the sort of bacterial groups that we want, so our sort of carbohydrate feeding bacteria. And then of course we've got all of our wonderful fermented foods like sauerkrauts and kefir and so on, to keep our guts nice and healthy. Okay, so another area that is known to be involved as a sort of inducer of nuclear factor kappa B is oxidative stress, so reactive oxygen species. And we know that high levels of sort of free radical damage and oxidative stress can target just about sort of every area of related health that you can really think of. So modulating free radicals and oxidative stress is obviously going to be another kind of target for intervention. And of course, there's an awful lot of research out there into the role of antioxidants and how they play a role in sort of modulating or reducing inflammation, or I guess sort of preventing inflammation in the first place. So we've got just kind of typical standards, anything through from things like sort of vitamin C to vitamin E, and there's a sort of plethora of sort of information out there and science, published science, that shows that increased antioxidant intake can have numerous benefits in regards to sort of reducing inflammation. And all of the products that I've kind of listed here really have been shown to reduce inflammation, specifically through nuclear factor kappa B. It's not the only pathway, but it is a pathway that's sort of relevant to what I'm talking about today. So CoQ10, for example, specifically as ubiquinol, I'll mention that a little bit more. There's a lot of science out there at the moment, and there's some very interesting science coming out specifically on ubiquinol. We've then got the sort of huge family of polyphenols, anything from resveratrol through to green tea extract, e.g. CG, and I'm going to expand a little bit specifically on resveratrol and curcumin in a moment. And then we've got things like sort of our carotenes. I was reading quite a lot about lycopene as well recently, but it seems to be that the evidence behind sort of lycopene that we see in sort of in vitro and in animal studies doesn't seem to come out so much or so strongly in human trials, and that actually the benefits of just sort of increasing lycopene intake naturally through sort of tomato based products, for example, is just as good for you. So tomatoes are rich, for example, cooked tomatoes and tomato kind of concentrates, so sauces and things like that, are even higher for some reason. Well, I guess it sort of makes sense really. But so yes, so sort of all these sort of dietary ways of increasing our intake of antioxidants is kind of important to sort of consider. And then we've got the wonderful sort of astaxanthin and zeaxanthins Astaxanthin, as you're probably aware of, is really kind of associated with krill and krill oil. And I would suggest that the benefits that are really associated with krill oil are probably coming from its relatively high levels of astaxanthin. So it kind of makes sense. Krill oil is very rich in phospholipid form of omega-3, not in high concentrations, but the phospholipids and free fatty acids are protected by this sort of high concentration of astaxanthin in there. It's quite interesting, actually, that a lot of the bioavailability studies that center on krill oil focus on sort of its unique sort of phospholipid composition, but actually there's an awful lot of free fatty acids in there. And because free fatty acids are so susceptible to oxidation, it kind of makes sense that there would be this sort of natural presence of an antioxidant. And astaxanthin is actually a really rather nice antioxidant on its own, purely because of its composition and the way that it actually sits within cell membranes is quite unique. And it makes it a very powerful and very effective anti-inflammatory antioxidant. But in regards to krill oil being a rich source of omega-3, that one I kind of had my doubts about. And for those of you who have sort of attended some of our webinars before, we've provided some quite detailed evidence to support that re-esterified triglyceride is in fact actually more bioavailable than phospholipids, and you get a much higher increase in the omega-3, which is kind of the purpose of taking an omega-3 supplement. Okay, so the nice thing about all these wonderful anti-inflammatories and antioxidants is that they are found in abundance within our natural diet. So so just to sort of give you an example, and this is really rather nice paper that I had a few weeks back, listing a lot of the kind of studies that have been published, looking at different sort of components, so anything through sort of like lycopene, lutein, and selenium, and all the sort of polyphenols, and you know the wonderful things that we find in things like garlic and mushrooms and onions and those kind of things. And then we've got a kind of a list down the right hand side of all the areas that are associated with inflammation. So nuclear factor kappa B, then we've got sort of apoptosis with the sort of cascades, we've got oncogenes, we've got tumor suppressor genes, and all of these natural antioxidants that we find within our food, that we find on our plates, have the ability to downregulate and upregulate where necessary all these sort of factors that are involved in modulating inflammation at the kind of molecular cellular level. And then another little sort of page here. So some of the ones that I've just kind of really been reading about a lot are sort of EGCG, resveratrol. Curcumin is a big one at the moment, and like I said, I'll go into a little bit more detail on that in just a second. So the sort of resveratrol, the wonderful red wine ingredient, the anti-aging molecule, as it were. Again, lots of really good supporting evidence for resveratrol in modulating inflammation through a variety of molecular pathways as well. Some of them are centered around nuclear factor kappa B, that are involved in sort of anti-metastasis, anti-angiogenesis, growth arrest, anti-inflammatory, and then we've got sort of other pathways that are involved in apoptosis. But resveratrol is incredibly good for sort of liver function as well. It's very good sort of for sort of carcinogen detoxification. As I said, without good, strong liver function, then you know we're not going to be able to deal with any kind of inflammatory load. And again, another rather nice list from a paper that was published in 2008, highlighting some of the sort of chemopreventative and therapeutic potentials or the mechanistic sort of perspectives of resveratrol. And again, and I haven't listed them all because there were sort of pages of them, but resveratrol, certainly in animal studies, in vitro studies, and some more recently in sort of human studies have shown that resveratrol decreases in most cases the production of sort of pro-inflammatory mediators. So again, sort of COX-2, so enzymes, COX-2, prostaglandins, that are all involved in eicosanoid, docosanoid production from long-chain fatty acids. And then again, so we've got our phase 2 detoxification and antioxidant enzymes. So resveratrol increases the expression of some really key and important detoxification antioxidant enzymes. So it's a good addition to. So I'm not necessarily telling you to go out and drink lots of red wine, but drink red wine in moderation is well known for a very long time to have numerous health benefits. But resveratrol as an add-on on its own has an awful lot of scientific backing behind it. And then from where IGenesis sit at the moment, ubiquinol is one of our favourite antioxidants. And the reason that we like ubiquinol so much is that it makes for an incredibly beneficial add-on for individuals who've got sort of energy issues. So we know that CoQ10, ubiquinol, plays a really important role in regulating energy production via ATP synthesis. But we don't tend to really kind of highlight the wonderful antioxidant properties of ubiquinol. Not only is ubiquinol a wonderful antioxidant, but it actually plays a role as an antioxidant recycler as well. So it goes around the body literally recharging various other antioxidants as well, which is really important because if you think about things like lipoic acid and vitamin C and vitamin E, once they've kind of donated their electrons to free radicals, they're kind of they lose their sort of effectiveness, as it were. So the fact that ubiquinol can go around sort of replenishing them, making them sort of good to go again, is incredibly important. And a lot of the time when we're sort of looking at sort of intervention studies, we see that the benefits of some of the sort of nutritional interventions tend to be supported when you've got more nutrients involved with each other rather than single nutrients as well. So we tend to recommend our ubiquinol as an antioxidant, certainly where there are high levels of inflammation, where we know that there is going to be high levels of oxidative stress. For those of you who are new to IGenesis or are not highly aware of sort of the bioavailability issues that sort of center around CoQ10 products, two versions of CoQ10, ubiquinol and ubiquinone. It's ubiquinol that is the antioxidant. It is ubiquinol that is involved in sort of the production of ATP. But whether you take either of them in supplementation, it's worth noting that they are both rather poorly bioavailable. Ubiquinol has the sort of key advantage of having a couple of extra hydrogen atoms, which means it's slightly more bioavailable than ubiquinone, but it still does have some issues. Around 60% of sort of CoQ10 supplements are thought to be excreted directly in the feces, for example. Because it's a large lipophilic molecule and it's fat soluble, it struggles to pass over the water, the undisturbed water layer that exists within the gut. So increasing bioavailability by taking with other fats is certainly required. Or you can take a ubiquinol supplement that has also the added benefits of having Vesisorb. Vesisorb is like a novel delivery technology that mimics the natural absorption process, improving the bioavailability of large lipophilic molecules. And this is how we ensure our ubiquinol product increases blood levels effectively. And so we've got a nice little pharmacokinetics profile here. Again, this is one of those kind of areas where there have been a lot of publications, and CoQ10, as ubiquinol, has been kind of like long standing in the literature. So we know that CoQ10 supplementation is incredibly beneficial. Again, there are certainly some sort of disparities between different studies. Some tend to say, yeah, CoQ10 is wonderful. Others will say not so wonderful. A lot of it boils down to bioavailability, and a lot of it boils down to how much ubiquinol gets converted to ubiquinol in the body. If you just supplement directly with ubiquinol, you will find that the benefits that are attributed to it as an antioxidant, as an energy-producing molecule, are going to be far superior. So anybody who is considering adding a CoQ10 supplement to their regime should really be sort of looking at the benefits of using ubiquinol over ubiquinol, and certainly sort of adding a bit of Vesisorb is going to ensure that your blood levels are at their very highest, because it's not necessarily always about how much you take, but it's about how much gets into your blood system. And this is just a little study that I've included here. This is very recent, published this year, basically highlighting the fact that ubiquinol is not only more bioavailable than ubiquinol, but actually has the capacity to reduce a number of key inflammatory markers. So monocyte chemotactic protein, for example, and also C-reactive protein. So that's our sort of marker. C-reactive protein generally is a marker that there is an inflammatory process actually happening. And the authors of the paper actually kind of go on to state that those with lower ubiquinol status tend to have, or a higher BMI, low-grade inflammation, would certainly benefit from ubiquinol supplementation. And some other sort of recent progresses when it comes to looking at ubiquinol over ubiquinone is in the area of Parkinson's. In fact, there's a lot of interest in Parkinson's and ubiquinol. It's been shown that there is issues with CoQ10 in Parkinson's, and some of the earlier studies suggested that Q10 may be beneficial, certainly with those with Parkinson's disease. The early research actually had so much backing behind it and created so much interest that it went on to sort of phase 2, phase 3 clinical trials. But some of these trials actually showed negative results, to the point where trials was because the CoQ10 levels increased, there was worsening of some of the symptom severity. More recent evidence has shown that actually the reduced ubiquinol form may offer benefits to Parkinson's patients. So I think those clinical trials are probably back on track, but highlighting and using ubiquinol rather than ubiquinone. And this is, for those of you who don't know our ubiquinol, this is our ubiquinol UH. You get 100 milligrams per capsule. And we add our Vesisorb so that you get unprecedented bioavailability. Because it raises blood levels so effectively, you can actually use it as a one-a-day dosing quite adequately. Okay, so I'm going to chat now a little bit about one of our sort of favourite subject at the moment here at iGenus, because we are incredibly interested in adding curcumin to our product range. We've been doing a lot of R&D recently, and this is one of the key products that seems to be really kind of coming out in the science community with extremely high backing behind it. And if you look at various sort of papers that sort of highlight the active pathways that curcumin is involved in, and again, it's sort of one of the areas certainly with there is a high level of interest is in the area of carcinogenesis. And if we just look very briefly at sort of curcumin and its role in regulating various sort of growth factors, inflammatory cytokines, transcription factors, enzymes, and various kinases that are all involved in regulating carcinogenesis and the various pathways. And a lot of these, again, are sort of focused through curcumin interacting and modulating the activity of nuclear factor kappa B. So curcumin is one of our sort of favourite subjects. It's a hot topic here. And again, a little similar to sort of CoQ10 and various other sort of ingredients or nutrients, if you like, issues surrounding sort of absorption and bioavailability have clouded some of the sort of curcumin research papers or the research studies. The issue really, when we're looking at sort of studies in regards to sort of absorption, transportation, assimilation, and elimination, tend to show that curcumin has low absorption, it's rapidly metabolized, it has a short half-life, and it leads really to sort of relatively low bioavailability. The other issue is that after absorption, curcumin undergoes conjugation, and the sort of main metabolic products that are derived from curcumin tend to have less activity compared to curcumin itself. So when it's metabolized, the activity of curcumin seems to be lost. And this is the sort of key area that a lot of the sort of research on increasing bioavailability of curcumin has sort of centered. And again, I sort of came across this paper some time ago now, and sort of doing some earlier R&D, which is a sort of the nanotechnology that is applied to curcumin. It's a summary of, as it were, for different sort of disease therapies. And you can see there's various different ways that have been looked at to increase the bioavailability and the effectiveness of curcumin. And the majority of the focus on sort of the development of sort of nanoparticles, the more sort of dominant ones tend to be sort of lysosomes and micelles. And this has led to the development of a number of approaches to increase and improve the bioavailability of curcumin. So we know that if you add black pepper extract to it, increases the bioavailability and influences things like half-life and so on. We can include things like volatile oils that are known to improve the bioavailability, but it's the nano formulations, so it's things like the liposomes and micelles, and to some extent the phospholipid complexes that appear to have some really nice sort of science backing behind them in regards to increasing the bioavailability and actually the activity of curcumin. So again, it's one of those kind of key areas that it's not just about taking a product, it's about taking it in a form that's actually going to have some sort of degree of activity. And again, numerous trials have centered around curcumin, and I've included this really because it kind of highlights some of the kind of key areas that curcumin sort of focuses on. I mean, they're all. This is a paper that sort of highlights the activity of curcumin in various different types of cancers, different types of doses, and you'll notice that generally the doses tend to be relatively high. So anywhere between sort of 1 gram up to 8 grams daily, it's quite high. But these also highlight some of the areas that have been established to be issues. So, for example, there was a phase 1 trial here that addressed the issue of sort of low bioavailability of some of the oral doses, whereas others have shown that doses of anywhere between sort of 0.5 gram and up to sort of 3 or 4 grams a day influences things like prostaglandin 2, sort of our pro-inflammatory prostaglandins derived from arachidonic acid. And then other studies have shown areas such as sort of tumor necrosis factor, again, interleukins, and things like oncogenes and various other sort of cancer promoting or sort of promoted genes as well. So yeah, sorry, got lost a little bit there. So phase 1, phase 2 trials here showing that this one was quite high actually, 2 grams up to sort of 12 grams a day, which is quite high, with black pepper extract. Well tolerated, improved bioavailability, decrease in things like nuclear factor kappa B, cyclooxygenase 2, and STAT3 as well. So the importance of looking at sort of curcumin products that have the best bioavailability is really, really important. We are currently looking into curcumin as I said earlier, to sort of add to our omega-3 stacks to complement our anti-inflammatory protocols. Because, as I said, quite often it's not just a case of one single nutrient is going to sort of be the answer to all of our issues when we're looking at sort of inflammatory regulation. But I can't leave the webinar today without having a little bit of a chat about omega-3s, so, and some of the sort of ways that they actually influence the interference of various sort of transcription factors as well. So if we just focus for a moment on our peroxisome proliferator activating receptors, these are a family of receptors that are involved in primarily regulation of glucose and lipid metabolism. But they also, through various different other pathways as well, so we've got our transcription pathway here, are involved in regulating various transcription factors, namely nuclear factor kappa B, to repress the production of pro-inflammatory genes. Omega-3s are known to be specific ligands for PAARs, and it is thought that omega-3s, some of the benefits that we see in regards to things like triglyceride regulation, body mass regulation, increased fatty acid oxidation, things like that, are to do with omega-3's involvement with PPARs in both of these sort of areas as well. So increasing PPARs and decreasing our pro-inflammatory genes, and increasing things like insulin sensitivity, regulating sort of triglycerides and various aspects like that, sort of cholesterol as well. And one of the ways, or one of the ways that omega-3 regulate nuclear factor kappa B is through PPAR regulation. So omega-3s are known to interact with PPARs, and there is just a sort of like little diagram here to show you various ways that omega-3 are known to interact, which then has this kind of knock-on effect at regulating nuclear factor kappa B, which then goes on to, okay, bye-bye, Natasha, to regulate nuclear factor kappa B. So we can down-regulate the expression of membrane receptors, so sort of cytokine receptors, nuclear factor kappa B, TNF-alpha, and interleukins, or we can directly increase the expression of our sort of inhibitory protein here, so we latch on to nuclear factor kappa B. We can inhibit the translocation of nuclear factor kappa B to the nucleus, so we can just stop it from actually interacting and influencing our pro-inflammatory translocation. Or we can interfere with the activation of transcription initiation complex itself here. So increase your omega-3s and you will decrease things like triglycerides, you will decrease cholesterol, you will have favorable effects on things like insulin sensitivity. And again, all of these are sort of involved in these wonderful molecular sort of pathways that are all sort of linked with each other. And so really sort of coming towards the end of our webinar today, there are many inflammatory regulating pathways that are involved in things like cell proliferation, differentiation, apoptosis, that are modified by various components of our diet. And again, it's not necessarily always about just one nutrient, but a combination of nutrients. And they have various sort of effects on things like plasma membrane function itself, or through to sort of the impact that we have on things like growth factor receptors, which then go through this wonderful cascade of abbreviated letterings. And then we've got the sort of death receptor. So this is sort of like the apoptosis sort of area. Growth factors are involved in sort of differentiation, proliferation, and so on. And here we've got our little friend nuclear factor kappa B at the bottom of here. So I mean, I've kind of covered a very small amount, and we're probably going to be having some more webinars coming soon that will be going into a little bit more detail about some of the other pathways that are involved and how we can target them. But what is kind of really key is that I think we forget sometimes how complex the whole inflammatory process is, and that inflammatory regulation centers on, interaction of different nutrients and interaction of different sort of key areas that are involved, and these all sort of intermingle with each other. So, anything from sort of cytokines to chemokine regulation to our toll-like 4 receptors and how we can sort of juggle these, how we influence sort of adhesion molecules, and so on. And we've got our nuclear factor kappa B here. So it's really kind of important that we sort of address inflammatory regulation from a sort of multi complex sort of interaction of different sort of molecules and nutrients, not only just through diet, but actually through a sort of variety of health supplements as well, where diet alone is known to sort of maybe not be quite enough to sort of help regulate. And so at the moment, this is our product range. The majority of you will be sort of familiar with our sort of therapeutic range, anything through from sort of Pharmica, Restore and Maintain, which is our sort of key protocol in regulating inflammatory balance. We've then got our neutral, a number of sort of additional nutrients. So we've got our NeuroBalance, our Super B complex, which we launched not that long ago, which is working incredibly well. And our Vesisolve ubiquinol. Of course, we focus on key areas such as bioavailability and absorption. So all of our products, we utilize the best ingredients so that you can be confident that your clients are going to get the best possible sort of therapeutic intervention. We've also got a wonderful well-being range. We are expanding our product range, so we will be introducing a rather nice multivitamin shortly to our product range. We're also introducing a new Pure Essential range, so our multivit will go into our Pure Essential range. We're also launching a sort of a general health high EPA DHA RTG, the esterified triglyceride EPA DHA product as well, to our sort of 1 a day health range. But then we are beginning to look at other sort of components or nutrients that are going to add value to our anti-inflammatory protocols. So we are primarily looking at curcumin at the moment, and we're also looking at various probiotics as well, given that they tend to be associated with so many different sort of areas of sort of health. If you would like to know anything more about any of the products that we already manufacture, or any products that we are sort of considering, or if you've got any feedback at all about areas that you're interested in, feel free to get in contact. Sophie Tully is our head of education, I'm head of technical. And of course, if you want to follow me on Twitter, please do. We try to sort of get as much sort of the latest research out there as we possibly can. And seeing as it's gone 1 o'clock, I'm going to suggest that if anybody's got any questions, if they'd like to ask, please do. Otherwise, feel free to email us individually. Otherwise, keep an eye on our list of webinars on our calendar, and I'll look forward to seeing you all, or seeing you all at our next webinar. Thank you very much, everybody.